It's biology with Mr. V. Biology with Mr. V. That's me! Hello, and welcome to a revision video about the GCSE topic, transport in cells. So I've darted around the outside of my title, the key areas, key concepts from this topic. We've got diffusion, factors that affect the rate of diffusion and this transport, examples of exchange services that diffusion takes place over, osmosis, the osmosis required practical for the GCSE, the potato pag as I call it, and the idea of active transport. So to get straight into it, the very first thing I'm going to do is actually make a little comparison between diffusion and active transport. So let's do that line down, that's not even remotely the middle, is it? Of all the things that could go wrong, I don't know what the middle of my pen is. And now I'm going to overthink it. I think, there you go, that will have to do. So diffusion this side. Active transport. I'm going to do it in a red pen. Active transport for me, I think right now I'm feeling red. I'm feeling that energy involved. So diffusion. I know that diffusion is the movement of molecules. Molecules move when they diffuse, that's what we mean. And I know active transport is also moving molecules. Look at that, guys, we've got a similarity. I know with diffusion that movement of molecules when it diffuses is how we would say it's down a concentration gradient, or to be more specific, the movement is from a high concentration to a low concentration. So if I sort of very swiftly draw what that could look like, if we have a high concentration of molecules on the left-hand side of this box and a low concentration on the right-hand side, diffusion would dictate that the molecules would move towards the lower concentration. Active transport, the reason why it's really good to compare, is the opposite. This is when molecules move from a low concentration to a high concentration. So again, if I do the same box approach, if I have a high concentration of molecules on the left hand side and a lower concentration on the right hand side, active transport would actually mean the molecules move towards the high concentration, which is completely against nature, really. It's completely against what usually should happen. Now, I don't want to get too crude, I don't want to give too many examples, but the key idea is, usually, molecules will always move from a high to low concentration. If you ever want evidence of that, spray some deodorant or some perfume in the corner of a room and just watch how that smell, the scent, those particles, those molecules spread away from where you originally sprayed the deodorant. If you left it an hour, the chances are, because those particles, those molecules will now be completely beautifully spread out all over the room, you might not even be able to smell it anymore. Active transport is the idea, basically, you spray your particles, then try and get them back inside the can. Very difficult to do. And that leaves us to the second sort of really big difference. There is no sort of energy required for diffusion to take place. Now, don't get me wrong, there is the natural kinetic energy that molecules have, but there's no extra energy. You don't need to respire and release energy for diffusion to take place, but you do for active transport. It requires energy from respiration. So here's a little pro exam tip for you. If you know a cell or a tissue is doing active transport, I can guarantee right now that those cells and tissues would have loads of mitochondria to provide the energy that they release by respiration, the energy to move these molecules against the concentration gradient. Right, let's have a look then, actually now, about cells and the sort of things a cell would need to increase the rate of these movements, to increase the rate at which molecules diffuse. So the first molecule I'm going to focus on is oxygen. I'm then going to look at glucose. You know, two rather essential molecules for all living things, because all living things respire aerobically, so they need glucose and oxygen. So I know if I want to increase the rate of diffusion, to increase the rate that molecules of oxygen and glucose get inside my cells where I need them to do respiration, I know that I want a larger surface area as possible. So if I want to increase the rate of diffusion, I want to increase the surface area. If I increase the surface area, I'm going to get a faster rate of diffusion. I also want any sort of barrier between the cells, which is usually the membrane, to be really thin. I want a really thin exchange surface to reduce a diffusion distance. You know, the less it has to travel, the quicker it's going to take, isn't it? I want a steep concentration gradient. The bigger the difference in concentration between the cell and outside the cell, the more molecules are going to move, the faster that rate's going to be. So in animals, the main way we have a lovely steep concentration gradient is a large and very good blood supply, because that blood is constantly taking away molecules and bringing new molecules. In the lungs, the fact that you're constantly ventilating by breathing in and out, that is also contributing to a steep concentration gradient. And last but not least, quite difficult to do with living things, but if I can increase the temperature, that means molecules have more kinetic energy. Just bear with it, I'm going to write KE for kinetic energy. Obviously in an exam, write it in full please. I overdid it, didn't I, with my spacing, so I'm just writing KE, but kinetic energy, please write in full if you're answering a question in an exam paper. If you increase the temperature, the molecules have more kinetic energy and therefore they're going to move more. So, let's see if I can draw an alveoli. I'm giving to myself because my drawing talents are usually not very well received by students or adults or anyone really. So let's roll. Here's my alveoli. This is the end of the bronchiole coming in, and I know an alveoli is like a little pocket, isn't it? It's like a little bubble. I think that'll maybe do. By the way, I'm not drawing a cell wall here, but I am drawing the alveoli wall. This wall, by the way, it's a layer of epithelial cells. And I'm desperately trying to draw it incredibly thin. I absolutely want a corresponding blood vessel, getting very, very close. It as well will have a wall on it, which will be incredibly thin. I'm just going to draw the wall on this side, the side where the exchange of oxygen and CO2 will take place. And obviously, no, I'm going to have to draw it on this side as well, aren't I? That's the OCD kicking in. Oh, that's desperately bad, but it'll have to do. So, as I said, this wall here is actually made of cells. Let's kind of demonstrate that by compartmentalising it. So is the wall of the capillary as well, so let's compartmentalise it to show that this is a wall of cells. It's not a cellulose cell wall, they are only in plants. Animals never have a cell wall, that's not the case. There you go, that'll have to do. Okay. So why is this exchange surface perfect for maximising diffusion of oxygen into the blood and maximising diffusion of carbon dioxide out of the blood? Well, number one, huge surface area. The fact that this ends with a large ball shape gives it a much higher surface area than if it stayed as a single tube. The bigger the surface area, the more spaces for molecules to diffuse. So we say in an exam, the faster the rate of diffusion. Secondly, these epithelial walls. They are 1 cell thick. So thin epithelial walls is giving that short diffusion distance. And thirdly, my blood vessel. The fact I've got a good blood supply. If I just sort of draw a few blood cells here. Yeah, it's supposed to be a blood cell. So's that. So's that. So's that. So's that. If I just show you kind of what the oxygen concentration, obviously the blood vessel coming in should have 0% oxygen. When it gets to where the surface of the alveoli is, some of the oxygen will start to move in. So say it'll start to pick up oxygen here. More oxygen by the time it's here as all the oxygen's diffusing through. And by the time we come this way, it should be completely full of oxygen. And likewise, the alveoli should now be empty. And because the blood is always moving, this oxygen that it's taking in is constantly moving away. So the blood that is always arriving at the alveoli always has 0% oxygen. What you're doing there is maintaining a steep concentration gradient. Steep concentration gradient. So these 3 points are essential for your exam technique to explain why any exchange surface is well adapted for diffusion. It will involve these 3 if you are looking at an animal. The blue parts are the descriptive points. The red parts are the explanations. The explanation must link to the correct descriptive point. If you say it's really good at diffusion because it's got a large surface area, you've got to therefore make the idea that because it increases the rate. If you talk about thin epithelial walls, you're only going to get credit if you explain it by it having a short diffusion distance. And so on, these 2 have to link together. There you go. Nice exam tip for you. Right, let's move on. Just really briefly, because again I'm trying to stick to a 20-minute time limit here. The other key exchange surface that they like to do in exams is the small intestine. Never forget, the small intestine isn't a flat tube like that. The small intestine has its large surface area because it has villi. Projections, folds of its epithelial lining. These villi, I'm just going to shade one villi in, that's a villi, that's a villi, that's a villi. They give it a much larger surface area, increased rate diffusion. The walls are one cell thin, just like the alveoli was, to give it a thin epithelium, so short diffusion distance. And again, the blood vessels literally line the outside, so a good blood supply to maintain concentration gradients. I will also stress, just before we move on, the small intestine does active transport as well. You will see a lot of mitochondria within the cells inside that villi. Active transport to ensure every last piece of nutrient that you digest is absorbed into your blood. Right, let's focus now on osmosis. I'm going to have to get the blue pen out. This is water, of course. So a definition. Osmosis is simply a special form of diffusion. So molecules are going to move down a concentration gradient. There's going to be no extra energy from respiration involved. This is a passive process. But it is only of water molecules. Water is the only thing that does osmosis, nothing else. So if your question does not involve water, it is not osmosis. If your question is water movement, it has to be osmosis. And the definition is always the same, and the definition is how you go about explaining observations. So my definition, the movement of, I'm going to put this in red because it's absolutely crucial, water molecules from a high water concentration to a low water concentration. Across a partially permeable membrane. I'm going to do that underneath, it's not very clear. Across a partially permeable membrane. We can split this into 3 marking points. 1 marking point for the movement of water molecules being identified. 1 that you're saying from a high water concentration to a low water concentration. And the final one is always a marking point for stating it is across a partially permeable membrane. Students and exams are usually very good at remembering this bit, because this bit can be a little bit tricky, the concept of high water concentration to a low water concentration. But they tend to forget the membrane part, which is always worth a mark. Which is really interesting because sometimes students misinterpret questions and they get this bit wrong. Even though they know it's osmosis, they might get this bit wrong by accident. Yeah, even if they put that just at the end, they would have still gained 1 mark out of 3. But they don't. So make sure you remember your membrane point. Absolutely crucial. Right, so in an exam, you are always going to be asked to apply your understanding of that definition to practical observations. And the most common practical observation is the potato required practical. So what we do in the potato practical is you get a few different concentrations of a sugar or salt solution. So I'm going to have a 0% sugar solution, a 10% sugar solution, let's do 20% and 30% sugar solutions as well. And let's put 20 mils in a beaker of each. Okay, so 20 mils of each of those solutions go into that beaker. I'm making sure I do the same volume of sugar solution because it is a control variable that's going to improve the validity of my investigation. To improve validity, I will also do repeats at each concentration, but that's going to be a pain to draw 3 different beakers for each. But I would do repeats to get that validity as well. So the other thing I'm going to add is I'm going to add the same size piece of potato to each. So let's say I added 2 centimeters by 2 centimeters cube of potato. So each one gets the 2 centimeter cubed potato. Okay, so it's got the same size, good for it. And all we do in this practical is we leave the potato inside the solution overnight. And you come back the next day and you look at the mass and how the mass has changed from before the investigation. So that would mean obviously you have to get the mass of your potato pieces before you put them in the solution, and then you get the mass of them after you put them in solution. So in this one here, if that's 0% sugar solution, well that's distilled water, isn't it? That's just pure water, no sugar. You'll tend to find that in that solution, the potato will gain mass. It'll get larger. And you'll tend to find in the more sugary solutions, my 30% one, that the potato might lose mass. Don't know why I've written loss there, it's meant to say mass. And you end up with a graph, if you plot this, that looks a little like this. So sugar concentration percentage there, I did 0, 10, 20 and 30. And actually, just to bear with me, I'm going to do this differently. Let's put the x-axis in the middle. So it's still sugar concentration in percentage. And I've still got 0, 10, 20 and 30. Gone a bit too far there, don't need all that there. So at 0 we said it's going to gain mass. So let's put change in mass percent. So if it gains mass, let's say it was there, and we said it was going to lose mass at the higher concentrations. Gives me a nice line of best fit like that. Obviously that'll be a nice straight line probably. Don't know what that kinky winky bit's about, never mind. Okay. Now firstly, there are 2 exam tips here. Number 1, very deliberately we are plotting the percentage change in mass. We plot percentage change in mass to account for different starting masses. Although you would have tried to control the size of the potato by keeping it the same, inevitably each cube of potato will have a slightly different mass. And to put it bluntly, you know, if I was to gain a stone right now, I'm currently about 11 stone, if I was to gain another stone, that's a 9% increase. If someone who is 7 stone were to gain a stone, well, you could say we've gained the same, but really, my 9% increase of a stone compared to their, 14-15% increase, they've gained more mass in proportion. So we look at doing percentages to account for any different starting masses there may be. So why has this one gained mass and why has this one lost mass? Let's take the one that we put in distilled water. The first thing for you to identify as the student, which one would have had the higher water concentration? Clearly, it's going to be the solution that is pure distilled water. So the 0% sugar solution has a higher water concentration compared to the potato. So water moves, this is my osmosis definition now, from a high water concentration to a low water concentration by osmosis. And clearly that means it's going into the potato. And don't forget the marking point, the fact it's going to be moving across the potato's partially permeable membranes. And therefore, the potato's now got more water inside it, which means it's going to have a higher mass than before. At these higher concentrations, it's the opposite effect. At 30% sugar solution, the solution would have a lower water concentration compared to the potato. So water will move from a high water concentration to a low water concentration by osmosis out of the potato, across partially permeable membranes. So it's the same answer, just reversed, depending on where the higher water concentration would be. Final exam tip before I leave you. They like to use this graph a lot in exams. And a very high level question they sometimes ask is, if we have this graph here, can you estimate the concentration of the potato? The sugar concentration of the potato? And the answer is yes, you can. The sugar concentration of the potato is wherever your line of best fit crosses the change in mass x-axis, where the change in mass is 0. The reason being is, at 0% change in mass, there's been no net osmosis. There's been no net movement of osmosis, no net movement of water. That's why it hasn't changed in mass. The only way that occurs is if the solution and potato have the same sugar concentration. You might use the term they're isotonic to imply that. Anyway, thank you very, very, very much for your time if you watched all the way this through. You are absolutely awesome. Take care, see you soon, bye bye.